Hi, my name is Sean Adams. I'm a professor in the Department of Surgery at ucdavis, and it's a pleasure to be here today to talk to you about the bioreactor in our gut. Before we start, I just wanted to disclose I am a founder of a company called Xenomet that is interested in this general area of the gut bacteria and the metabolites that they make. So, a brief overview about the gut microbiota, and many of you have perhaps already heard about this, but the bacteria in our gut, the number of bacteria in our gut are about the same number of cells as in our body, if not more. In fact, it's been estimated by some people that about 100 billion bacteria are in each gram of stool, if you can imagine that. There are about 500 times more genes in the bacterial genome than our own genome. And what this means is, as you might imagine, tremendous metabolic potential. Now, the gut microbiota have been associated with many physiological functions. We know that these bacteria and other microbes change in the gut due to a variety of factors. To a degree, food obviously affects the bacteria that are there and what they're doing, but also, interestingly enough, things like bariatric surgery, diabetes status, and even fitness. So physically fit individuals have a different microbiota behavior than folks who are more sedentary for reasons that we still don't quite understand. So many things go into modify the gut bacterial communities that are there and what they're doing. These bacteria have been associated with many physiological systems, health and function of the immune system, the GI tract, liver health, and kidney function, just to name a few. Other people are studying things like the gut-brain axis, where the belief there is that the bacteria are somehow influencing our mood, how we think about things, so the brain function can actually be influenced by what's in your gut. The big question is, how is this possible? How are these bacteria talking to the host? And how are we talking to the bacteria? Now, many folks out there kind of think about the gut bacteria in a different way, and that's through the use of probiotics or functional foods and fermented foods. Many of us are familiar with these. This is just a graph kind of showing the increase in the market value of supplements, including probiotics, that are in the many billions of dollars. But what's very interesting is, despite many of these probiotics on the market, it's really a black box in terms of the mechanisms of action, how these impact health and function in the body. And so understanding that can actually be very valuable as we start to really think about which probiotics should we use and should they be personalized to your specific situation. We'll really only get there when we understand what's in that black box. The take-home message, just to kind of jump to the end here really, cataloging the bacteria, knowing who's there, is largely insufficient to understand what they're doing and how they do it. And that's why today's talk focuses on Xenometabolomics, which I'll define here in a minute. And the outline of the talk will really be the case for Xenometabolomics. Why study this? What is this field? And what's it sort of good for? Second part of the talk will be the us versus them. In the bloodstream, there are probably 50,000 metabolites coursing through the blood every day. What percentage of those are actually from the bacteria in our gut? This is a very interesting question. It has profound implications for health. And then finally, I'll end the talk by talking a little bit about how xenometabolites themselves could be considered as bioactive in the body and think about potential links to health and function. So let's start with the case for xenometabolomics. What is this and why do we study this? So what is metabolomics? Metabolomics is a system-scale discovery or study of the biochemical intermediates in biological systems. Think of the bloodstream and think of when you go to a doctor. They'll often ask for lab tests to be done for cholesterol, blood sugar, these sorts of things. Well, these are just a few of the metabolites in the blood. It's thought that there are about 50,000, between 40 and 50,000 metabolites actually in the blood. So these things we typically have measured at the doctor's office are just a small fraction. So the study of metabolomics is really trying to take a snapshot of that entire chemical landscape in the blood or any other biological system. Now, xeno derives from the Greek xenos, which is alien, foreign, strange, or unusual. And so this leads to this term xenometabolite or xenometabolomics. And what that means simply is metabolites or molecules, say in the body, that don't come directly from us, but are derived from something that's foreign, say, for example, microbes and bacteria. Now, a dirty little secret, and the reason xenometabolomics is important, is the diet itself has a limited influence on the gut microbiota. Certainly, the diet and certain foods can impact the bacteria and other microbes that are in our gut, of course, but not as much as you would think. And I'll show you kind of a complicated slide that I'll try to make sense of here in a minute. So this was a very nice review paper published by Rob Knight and his colleagues. And those dots you see in this graph are really, these are each color is a single person. So blue is one person, red is another, and so on and so forth. And those dots are really mathematical scores which are derived from the variability in the gut microbiota. So if the gut microbiota changes due to diet or whatever, those dots will move for any given person. So if we look at the person, the red person, for instance, as they switch from a high-fat to a lower-fat diet, plant-based, animal-based diet, their microbiota does change some, but really they continue to look like themselves. They don't look like the other folks in the graphic. And the point here is simply, yeah, diet can impact your gut microbiota some, but really your innate bacteria are very sturdy. They tend to want to stay where they're at for your individual situation. And that'll be really important later when we talk about the metabolome. Yeah, another example of where the diet actually has modest effects at best on the actual microbial species that are there comes from this study here, which was actually a randomized trial related to the dietary guidelines for Americans, and sort of comparing that against a typical American diet in obese women. Now, the interesting thing about these diets is they were weight maintaining, but the participants in these diets were fed every bite, and every bit of food they ate for 8 weeks was monitored and known. And in the course of this study, there were fecal samples taken at sort of 1 week or right before the dietary intervention started, about 2 weeks into the dietary intervention during week 3, and about 8 weeks into the dietary intervention during week 9. And the question was, does this actually impact the microbes in the gut? And what was interesting, and this is kind of a complicated slide, but just to keep it simple, the community structure within any given sample can be measured through a variety of mathematical terminologies that sort of indicate alpha and beta diversity, in this case alpha diversity. And the two different colors here are the two different diets. And left to right is before, 2 weeks into the diet, and about 8 weeks into the diet. Each of the panels represents a different way to express alpha diversity. And I think what you can appreciate is that really it didn't matter. The diet didn't really change the alpha diversity of the community structure of these individuals. The beta diversity, which is across different groups, also didn't change. And again, these are just different ways to show this diversity score across all the individuals. Each dot in these graphs is an individual, and showing it from pre to 2 weeks to 8 weeks going left to right. What you can see, I think, and appreciate is there's really no difference in this beta diversity measure across the two diets either. Now, there were some species of bacteria that changed in the gut, and these are just a couple of examples shown at week 2 and week 8, where there were a few species that did change based on the diet, but not a whole lot. Now, the reason that this is really important is I'm going to try to convince you that what can change a lot in these different situations is the xenometabolome, or what the bacteria are actually doing, which is probably much more important than the exact species that are there much of the time. So let's think about inulin, which is a very common type of fiber added as a supplement to different foods. Sometimes people will actually take it as an individual supplement. It's a type of fiber that is thought to convey health effects to people. So it turns out, I'm going to show you that knowing what's going on with the metabolome is much more instructive than knowing what's going on with the community structure or the species that are there. And this was a collaboration done with Steve Lindemann at Purdue University, as well as Brian Piccolo and Ming-Su Chen at Taiwan National University. Now, the study design is pretty simple. There were 3 different donors, donor 1, donor 2, and donor 3, left to right, who were studied. The stool samples were taken from these donors, and they were treated with different kinds of inulin, different kinds of fiber. These are shown on the right, with these sort of funny terms, AA, L90, CLR, and so on. These are just different types of structural inulins that the bacterial cultures were fed for 7 days. And over 7 days, you might imagine, some of the community structures and some of the species would change around a little bit. But at the end, we also did Xenometabolomics analysis of the culture samples. Now, this again is one of these complicated slides, but it doesn't have to be. Left to right is day 0 through day 7 of these cultures. On the top is donor 1, in the middle is donor 2, and in the bottom is donor 3. And I guess the point is, for this particular kind of inulin from agave, I think you can appreciate that the different colors indicate differences in species or community structure, I should say, of these cultures. So the first thing that should jump out at you is that in fact each of these donors look very different from each other. So the community structure, the colors are very, very different between donor 1, donor 2, and donor 3. A different kind of inulin, in this case fructulose, shows you the same thing. Each of the donors looks very different from one another. The inulins can actually influence the bacterial species to a degree. But the key is each donor looks different. So how can we explain this, and how can we ever imagine that fiber is going to help anybody when we all look so different from one another? Well, the trick may be that it's the metabolome that's most important. So this particular graph shows for each of those culture samples a score, and this score is a mathematical score which is based on the metabolome, the variability and differences in the metabolome for each one of those different conditions. And I think what stands out right away is that the agave inulin looks very different in terms of its metabolome compared to the other ones that were tested. Now, this is interesting because, of course, the communities were very different. All 3 of these donors were very, very different from one another. But when it comes to the metabolites, they all begin to look very similar under this particular treatment regimen. This is another way to look at it here. It's called a heat map. And along the right-hand side there are, in tiny little letters, those are individual samples that were tested. And along the top, these are different metabolites that were detected. And we don't have to know which metabolites or samples. I think you can appreciate that when something is more red, that means it's higher in that particular condition. So despite tremendous community differences in terms of species and phyla, the metabolome looks very different within any given inulin. And you can begin to actually tell the inulins apart from one another by looking at things in this visualization here. So here's the way I like to think about it, as a functional guild. So it turns out there are bricklayers. They're sometimes called bricklayers unions. Sometimes they're called bricklayers guilds, as shown in the upper. And I think what you can appreciate is looking at this picture, these 2 pictures here. All of these gentlemen look very different from one another. Some are tall, some are short. They come from all kinds of backgrounds. But what's interesting is they're all trained to do the same sort of thing. They have the ability to do the same sort of thing, to create, working together, a seamless, beautiful building. You'd never be able to tell which of the gentlemen on the left helped to build that wall. They all did. And this sort of guild way of thinking is how we're now starting to think about it metabolically for the bacteria in our gut. That we can all have very different bacteria in our gut across the population, but we might respond very similarly to certain fibers and certain foods at the metabolome level. So knowing the metabolome is very, very important. Us versus them. This is a fun part as we get into the middle of the talk. Out of the 50,000 metabolites in our blood, how many actually come from the bacteria? And this is usually where, if we had a live audience, I'd have everybody vote, but we're not going to do that today. So us versus them. How do you study this? A very difficult thing to study. One way to do it would be to compare the metabolites that are in the cecal contents, or the large intestinal contents, or the feces, compare them to blood plasma. Those that overlap perhaps could be a sign of bacterially derived metabolites that are ending up in the blood. So here's a study we did a few years ago in rats, where we looked at the cecal contents, the lower GI contents, and compared them to plasma. And we found about 40% overlap, meaning the blood contained about 40% of the metabolites that we also detected in the large intestine. Here's a more recent study. We didn't do this study, this was by a different group, but it was in mice and looking at metabolites again that were different in the feces, or pardon me, that were shared in the feces and the plasma of these mice. And strangely enough, they also came up with a value of about 40%. Now, this is a kind of a complicated slide, but this is what people are doing nowadays, and that's to do correlation statistics. So if you know the bacterial phyla that are there, or the species that are there, you can then correlate them statistically to the metabolite levels in the blood. And a Spearman's correlation statistic is used here. And the little dots in the middle, these are in humans, by the way, the dots represent the correlation statistic of any particular bacterium in the stool compared to any particular concentration of metabolite in the blood. And the 0.25 numbers mean about 25% is the way I could think about it, and would encourage you to think about it. About 25% of the variability in the metabolite could be explained potentially by bacteria, specific sets of bacteria. You can winnow it down a little bit to be 0.125 or 12%. So I know this is complicated, but the point is, one way to do it is to say, hey, look, if a bacteria is increasing in its abundance in the gut, does the metabolite increase as well in lockstep? Let's do a correlation statistic between the two. Another way to do this is to look at germ-free mice. Now, germ-free mice have no gut microbiota at all. So you might imagine metabolites that are lower in the blood of these mice might be candidates for xenometabolites from the bacteria. So this group here did a pretty simple study where they looked at germ-free mice, or specific pathogen-free mice, which are sort of controls, and they went and got the feces and looked at differences in the feces and in the blood for metabolites between these two. And the long and the short of it's shown here, which is basically if you look at the number of metabolites that are significantly reduced in the germ-free mice compared to the wild-type mice, it's about 12% of the entire metabolome in the blood can be explained in this way. Sounds very familiar to that slide I just showed you with the statistical correlation. This is a little hard to read, but this is just another paper that would suggest that it sort of depends on how you read it, but between about 15 to 35% of the variability in the blood metabolome could be explained potentially through the microbiota, was their conclusion. So what is the actual number? Well, I'll tell you that in a minute, but according to my opinion. But there are many interpretive challenges to figuring this out. First of all, stool or feces is typically measured, but this doesn't reflect differences in bacteria that occur throughout the many meters of GI tract tissue. So we really don't know what's going on in the upper gut when we're looking just at the stool or the feces, making it really problematic. Bacteria values are often expressed in relative percent terms rather than actual numbers. This makes correlation statistics very tricky. We often look at a particular pool, like a urine or blood pool, but we really don't know the flux or how these metabolites are moving over time. We just know a spot in time for these concentrations, again, making it very difficult to interpret this. The gut lumen metabolite concentration is usually not measured. We don't know what the concentration of most of these metabolites are actually inside the gut. We're relying very often on, again, feces or stool to make these calculations, so it's very difficult. And then finally, when we're trying to correlate specific bacteria to specific metabolites, that guild thing I mentioned to you earlier with the bricklayers guild comes into play, where maybe looking at specific bacteria with specific metabolites is the wrong way to go. We should be looking at groups of bacteria that are able to do the same thing metabolically. Here's an example of what I mentioned earlier, which is sort of the differences along the gut for not only the bacteria that are resident there, but also potentially the metabolites. We don't typically have the ability to interrogate the metabolites and the bacteria that occur that are differentially abundant throughout the gut of any given person. So this makes it really difficult to consider some of these questions. Now, more recently, there's hope. There's some great work out there shown here and highlighted in which this new technology called a CAP scan has been used, which is a capsule that you could take, and it can interrogate, and it opens up and samples at different parts of the gut and then can be retrieved later and measured for metabolomics. And this has really been revolutionary in our ability to understand what's going on throughout the gut. Here's an example. Again, these are mathematical numbers, which are basically representing the variability in the metabolome of any given sample from any given person in any given condition. And I think what you can appreciate is the pink oval toward the right looks very different from the other ovals on the left. Well, turns out the one on the right are the metabolites measured in the stool, which is where we normally get sample. The ones on the left are from these devices at different parts of the gut. So immediately it says that the metabolite profile in the stool does not reflect what's going on bioregionally. But there's hope. I think we know the answer to the percentage of the metabolites in our blood that are coming from bacteria. We don't know the exact answer, but I think we're getting closer. My conclusion is that at least 20% or 1/5 of the metabolites in the blood are coming from bacteria. And that's using all that literature I just told you about. Now that's remarkable because if there's 50,000 metabolites in our blood, 10,000 of those are from the gut bacteria. So what you're eating, whether you're physically fit, whether you have diabetes, a whole variety of factors go into play to modify the xenometabolome. That in turn can modify very dramatically what's in your blood. I'll end with some bioactivities. So why do we care? We can catalog all these bacteria, we can catalog all these xenometabolites, but at the end of the day, does it really matter? Do any of these sort of 10,000 plus metabolites have any bioactivities in the body that could link to health? Here's just a couple of examples. We've been studying this class of metabolite in our lab called the cyclopropane fatty acid xenolipid class. And there's an enzyme shown in red there that's only found in bacteria, not found in us. These bacteria can take normal, sort of everyday lipids that we eat in the body, such as olive oil, and they can convert them, at least in theory, to these cyclopropane fatty acids shown there on the right. Well, what's interesting is these cyclopropane fatty acids can bind this very important receptor called PPAR receptor, which is sort of shown in these ribbons here, and the yellow part is the cyclopropane fatty acid kind of binding and embedded within this protein. These proteins are critical to regulating metabolism in the body, so you could instantly begin to think about, wow, maybe the xenolipidome could be influencing our metabolism by binding some of these receptors. Some of these metabolites also influence inflammation. So endotoxin is a pro-inflammatory component of the bacterial cell wall that can actually lead to sepsis. It's often used in cell culture studies like shown here as a positive control. It can induce inflammation in macrophages, these immune cells. So if you look left to right, and this is just a readout of a particular cytokine expression here as a marker of inflammation, you can see that the endotoxin or LPS activates inflammation very profoundly as expected. Now when you add in these unique xenometabolites, either as a pool, a mixture 1, mixture 2, mixture 3, or as individual metabolites in increasing concentration shown in those blue triangles, you could see an immediate blunting of that inflammation due to LPS. So even in the presence of something that's known to cause sepsis and inflammation in the body, some of these xenometabolites actually have pretty profound anti-inflammatory effects. This is another example. You don't need to know exactly each of these boxes. I can tell you that each one of them is a cytokine. Each one of them is a marker of inflammation in each of these panels. The highest bar shown there is, again, endotoxin in these macrophages. So you can see endotoxin increases pretty dramatically the inflammatory marker. But as you increase the concentration of the amino acid tryptophan-derived indoles, shown there as you go left to right, you can see a blunting of inflammation, which is pretty profound. So again, another example where these xenometabolites could have potentially very profound effects on our health. Let's end with some future directions. Where are we going from here? Well, we can begin to think of targeted modification of specific xenometabolites through diet, foods, supplements, prebiotics, and probiotics if we knew which xenometabolites we would want to affect in any given situation, we can now begin to think about how to deliver them directly to the GI tract, or to have the gut bioreactor that's already there produce these for us. Here's an example of one study that we're doing now with almonds, called the Healthy Unique Lipids in Almonds study, or HULA study, in which we propose that the monounsaturated fatty acids of the almonds, which are considered to be very healthy, may be acting in part through conversion to these xenolipids by the gut bacteria. So we're testing that now in our laboratory. We can also think about biomarker screens. What if we actually had screens where when you go to the doctor, they're no longer just ordering blood sugar, cholesterol, triglycerides, and other things? What if they were able to interrogate your gut microbiome metabolic phenotype? That would be very exciting. Something we published a few years ago called the Xenoscan begins to think about this, where you might end up with two different people. On the left is a healthy person, on the right maybe somebody with type 2 diabetes, and their microbiota are doing very different things metabolically. So we refer to that as a metabotype. So on the left you might imagine a microbial metabolite A, and on the right a microbial metabolite B. So what if we were able to take a single blood sample and understand exactly what's going on in your GI tract with respect to the microbiota? And we maybe don't even have to know the species of bacteria that are there. We would care more about the metabolism and what they're doing. And what's interesting, just as a proof of principle, these are some papers shown here that are examples. Just having type 2 diabetes alone, all other things controlled, actually impacts your xenometabolome. So somehow having diabetes, being diabetic, somehow influences the bacteria so that their metabolism actually shifts and changes to create a unique Xenometabolomics signature. That's very interesting. So the take-home messages. Cataloging the bacteria are really, this is very important. Knowing the species, knowing the phyla, very, very important. But it's really insufficient to understand what they're doing and how these different situations are linking the microbiota to health. Metabolic functional guilds are probably major players in the diet microbiome host nexus. So individual bacteria, again, very important, but it's probably more these functional guilds that are impacting how the bacteria and the microbes are influencing our health and function. Now, xenometabolism R&D helps us to get the full picture of host phenotype and microbial metabolism, helps us understand how microbes talk to the host and vice versa. And it's also very important to think in the future, how we might harness these metabolites or these processes for nutraceuticals, microbiome-based therapeutics, and even biomarker development. That's actually the next frontier, and the next gen of probiotics and prebiotics and so on will be in this space, which is exceptionally exciting. So with that, I want to thank everybody for spending time with me today to go through this kind of unique and emerging field, and want to thank my entire team of my laboratory shown here, especially Trina, Jean, Jordan, and Lorena. And again, our funders are shown on the bottom. We thank them very much. We wouldn't be able to do what we do without them. And collaborators are also very important to what we do. There's no way we could do it without their help. So thank you very much.